Okay, great. Hello, everybody. Good morning in this beautiful country and in this with this beautiful weather. We didn't know the weather, so excuse us. Hope everything will be much better than the weather. Welcome you all in our kickoff conference of the LIFE project Apollo 2020. I would like to invite Director of the Karkonoski National Park, Pan Andrzej Raj, and to welcome you and open the conference.
Good morning, everybody. The weather of Karkonosze, it is typical weather for Karkonosze Mountains. That's, you should be surprised. Okay, dzień dobry. Good morning once again. Ladies and gentlemen, welcome to all of you in Karkonosze National Park, particularly in this historical building of Szafgocz family, where we are adopting for the education center. It is my great pleasure to see all of you at our conference, Science, Ecology and Innovation for Parnassus Apollo Conservation in Central Europe. The Apollo 2020 project brings together scientists, park administration managers, NGOs, and other stakeholders involved in the protection and restoration of the Apollo butterfly population in Poland, Czech Republic, and in Austria. Today, we are here together to discuss, exchange knowledge, exchange experience, look for new inspiration for better and more effective protection population of Apollo butterfly. I am very happy with the number of conference participants from Poland and many European countries. And I wish you very good discussion, achieving the goals of the conference and achieving the best results. Finally, I wish you good staying in Karkonosze National Park, Karkonosze Mountains. And we, all my colleagues from Karkonosze National Park, declare help and friendly atmosphere during the conference. Thank you very much and have a good stay in our new education center. Thank you very much.
From all this stress, I have forgotten to introduce myself. I'm Magdalena Makowska, and I'm from National Naturalist Club, and I'm a project manager of this project. And I will be your host and moderator for today. Some technical things. Those of you that use the simultaneous translation, I hope you already know, but if not, the channel 1 is for Polish, channel 2 is for English, 3rd is for Czech, and the 4th is for German. So if you haven't noticed yet. Yeah, so I think I would like to introduce our first speaker, Mr. Paweł Adamski, who will tell us about the species itself, the status in Europe, threats and biotop.
Thank you very much for the honor to be here and speak to you. There is often question, why Apollo? Because Apollo butterfly was in Europe the first. It was the first species mentioned in the CITES Convention. The first species which were mentioned as endangered. But when we look on the general threat status of this species in the whole geographical range, we realized that it's only the least concern. So in fact, it isn't generally critically threat. However, when we looked in the local scale in Europe, among countries which prepared a red list according to IUCN methodology, we found that it's much. Oh, I'm sorry. It is much more endangered in the Europe, in the particular countries, as in the general in its geographical range. One may argue that, in fact, this European endangered is not important because it's only kind of marginal area. However, including the Apollo butterflies in all important convention of protection means that in fact it's really important. Why? Because why Apollo butterfly is so unique? Because it's amazingly diverse. The most than 100 subspecies have been described among the Apollo butterfly. Of course, currently there were several revisions of this information. However, it's still a lot of great amount of the subspecies. Why this species is so diverse? There are two main sources of this status. The first one is history of the Apollo butterfly in Europe. The species originate from the Asia, current Mongolia, and in Holocene 2 times colonized the Europe. There was the first colonization, the species survived after this colonization in the Spain and in some west south localities, and there was second colonization, which finished the current distribution of the species. Moreover, the distribution of the species is very fragmented. When we see on the general map, we see this solid range. However, in more detail, we see that there is the main part and the more or less fragmented sites in Europe. In more detail, it's still more fragmented, and also it's only Carpathian. We see that there are several fragmented sites. Moreover, among these fragmented sites, these shapes mean the place with the same subspecies. We see that only in the Carpathia we have the several subspecies, in fact closed but isolated. This fragmented distribution and the history of the species may affect in the first stochastic genetical changes among the small fragmented population, founder effect and genetic drift, but also what is more important, local microadaptation to the local habitats, local condition. In result of this process, particularly this microadaptation, that the differences between the subspecies is not only pure alleles or allozymes differences. It also means differences in morphology. Here we are several examples of some subspecies. They differ in the shape of this ocelli, the shape of this. Generally, they differ in some morphological character, but also what is very important from the conservation point of view, there are also differences in the physiology. What does it mean? The first, it means different preferences of the host plant. There was analysis of Professor Nakonieczny from the Silesian University. They analyzed the 3 general groups of feeding. There were Albiphogus, it means the caterpillar, particularly the first stage of particular, feed only or mainly on the sedum album, and Telephophagus, there was the second group. There was the subspecies which caterpillar, particularly the first stage of caterpillar, fed only sedum telephium. Moreover, in this second group, which was very detailedly researched by Professor Nakonieczny, he found that, for instance, Pieninian Parnasius Apollo frankenbergeri, its first stage of the caterpillar may feed only one particular subspecies of sedum telephium. So it's very specialized, and there are several places when, in fact, there are not analyzed the feeding preference of the larva, or data are different. What does it mean from the conservation point of view? Firstly, the extinction of the local population mean that we lost the unique gene composition, unique for this population, local population, or maybe for the subspecies. Moreover, during the restoration project, we have to remember about the local adaptation, because when we try to introduce the individuals from the different localities adapted to different habitat, we may meet the problem of kind of maladaptation. The one of the possible maladaptation was that in some southern population, Apollo butterfly is bivoltine. So it means it has 2 generations during the vegetation season. The first one in the early summer, and the second one in the late summer and autumn. When we introduce such subspecies in the north, this second generation is unable to survive, and if there are not eggs waiting from first generation which survive the winter, it may be very dangerous. Moreover, when we try to improve the genetical quality of the introduced population by some kind of crossbreeding, there is the risk of the outbred depression. So, you know the meaning, the outbred depression. It means that we know that when we crossbreed the individuals which are too close, it's inbred depression. However, when we crossbreed the individuals which are very far from one another, we may take the outbred depression. For instance, we cross the Albivorus with Telephiovorus. We hope that it will be omnivorous, but in fact, it may be nothingvorous. So So it's an example of outbreed depression. But why the Apollo is so diverse and so endangered and so important? The second reason of unique importance of Apollo butterfly is so-called umbrella effect. Umbrella effect of the whole habitat. In Europe, Apollo butterfly inhabits the alpine meadows in general and pastures. This type of habitats are very often endangered. They're decreased. Moreover, there are a lot of invertebrates connected with such habitats, which are not so visible, so flagship like Apollo butterfly. What is also important, Apollo butterfly is very sensitive for the habitat changes. So, when Apollo butterfly decrease, it means in the short perspective also another subspecies of the animal and plants typical for these habitats may become endangered. So, if Apollo is good, we hope another species is also in the good condition. Moreover, advantage for using Apollo as this umbrella effect, it's well visible, easy to monitor, and also it has very positive social image. So people are not nervous. Why we have protect an ant or something like that? We have protect the butterfly. Huge, interesting butterfly. There are some picture from the habitat. As you see, there is usually very fragmented. It's a Pieniny mountain, it's Podskalnia Góra and Spuszczalnica locality. But why it's threat? What are the threat factor for the Apollo? The first one, there are in fact two groups, direct and indirect. The factors directly affecting the species are predators. Apollo butterfly is predated by the birds, by the spiders. You see the Apollo butterfly in the spider web. And also the most dangerous predator in Pieniny mountain are the ants, which are really very, very effective in killing and eating Apollo butterfly. The collection. It's a very beautiful butterfly, so people like to collect them. And there's a still continuous problem with illegal collection of Apollo butterflies. The host plant decline is usually connected with the habitat changes. I will be talk about it for the few minutes. Pesticide. There are some information that overusing the pesticide can affect the Apollo butterfly population, and of course, genetical erosion. In the small population, some kind of inbred or another genetical problem may occur. There are some examples from the Pieniny mountain. There are individuals which weren't able to stretch the wings after clutching from the pupa. There is the individual with the rest of the pupa's fragments on its Right? And also there are kind of the weather catastrophes in the during stress. And there are when there's also the individuals which are unable to fully stretch these wings. But there are also factors which are affecting indirectly the Apollo butterfly. The most important factor is the habitat changes. We cannot maintain the butterfly population when habitat is lost. So, let's look for the example of Penindi Mountains. The people they really job, they compare the same places in the 20s and 30s of the last century and the current situation. We see that there is now the mountain is almost fully afforested. The habitats suitable for Apollo butterfly almost vanished, and it's the main threat factor affecting this species. This change of the landscape is maybe human drive, like, for instance, changing in land use, particularly afforestation, but also it may be natural or semi-natural, like ecological succession. There are also several factors which affect Apollo butterfly both directly and indirectly. The first one is long-term climatic change. This species has evolved in the cold steppes. And now in Europe, the similar conditions are in the mountain. But during the climate warm, this condition change. Also, one of the effects of the climate change is the increase of the weather catastrophe, like rapid rainfalls or flash floods like that. And Apollo butterfly, in the few hours after hatching from pupa, is extremely sensitive. When there is the heavy rains in this time, it is unable to fully stretch its wing. How to divide? One might ask, how to divide this not fully stretched due to some bad condition of individuals from not fully stretched due to weather? There are some symptoms. For instance, for example, it is this breaking of or not fully stretching of wings. It's typical that the individual was strength enough to strengthen these wings. However, when they was very soft, there was change, and it can't be, and it is permanent. Moreover, if there are some, because butterfly stretch their wings in the due to its importance. So firstly, the forewings are stretched, later the hind wings. If there are bad weather conditions, sometimes butterflies are able to stretch only the forewings. Of course, habitat fragmentation. It also affects the species directly as well as indirectly, and air and soil pollution. So, what is in my opinion, the ultimate conservation goal for Apollo butterfly? At the general level, we would like to conserve the unique diversity of the species, this great amount of subspecies, its diversity. Or some people think that maybe we should restore the species in all plausible species. I'm not sure it's a good idea, in fact, but there is also some goal. And at the local level, we may maintain or restore viable, self-maintaining population. But if we can't do it, also we have maintained or restore plausible metapopulation structure, because in the metapopulation, if there are too much distances between subpopulation, in fact, we have no metapopulation, but some isolated subpopulation which are prone to the genetical drift or inbred depression. Sometimes, in fact, the best what we may achieve is to create some kind of so-called wild captive metapopulation. And of course, we have to maintain or even restore the Apollo habitat. Thank you for your attention. Any questions?
Thank you very much. Are there any questions from the... No. Okay, so then thank you. And yeah, I would like to invite our second speaker, Mr. Gerard de la Vera.
Okay, waiting for the slides when it's possible. It's just, I'm waiting for the slides to appear, so should I just... Well, anyways, maybe I can start before the slides start. Just thank you for the introduction and for the invitation to come here. Actually, it is fantastic to be here and to be part of this conference, this unique experience of conservation with one butterfly species. Here it goes. Okay. So first thing I would like to say is that I'm actually not an expert on butterfly conservation genetics, and meaning that I don't have like direct hands-on experiences on taking genetic-based decisions for conservation programs. But on my defense, maybe I would say that I don't actually know anyone who actually have such hands-on experiences on taking genetic-based decisions for conservation programs in butterflies. So in this sense, I want to congratulate the partners of this LIFE project, because I think this is a very good opportunity and very for their determination actually to have this broad perspective in a conservation of one particular butterfly species. So it is my intention to drive you through a review of what is the current state of conservation genetics in general, but most particularly in butterflies. Indeed, conservation genetics it's a pretty recent or not very new field of research, especially applied field of research. And as you well know, biodiversity it's experiencing important decreases, losses of diversity, mostly because of human pressure. And this is not new. This is something that was actually already aware by classic naturalists as Wallace, for example. So biodiversity is a suite of complex processes that happens spatially and temporally, and in these processes we have These processes just lead to the diversification of this diversity, so we gain diversity on one side, but these processes also affect diversity in a detrimental way, so we also experience losses of diversity that ultimately leads to extinction. And these processes is what we evolutionary biologists study. And while these processes in evolutionary ecology inform us about what is going on out there and why we are losing or potentially losing biodiversity, this information makes little sense or is not enough for our conservation goals if there is no application after this information. So in this sense, evolutionary ecology informs us, gives us the information, but conservation biology protects these potential losses of diversity. And I think we mostly would agree that in the field of ecology and evolution, there has been a historical disconnection between these two fields, in the sense that the ecology and the evolution of the species has mostly been studied quite apart historically, and this has led to some kind of disconnection in between these two fields. But we need to consider that ecology and evolution needs to be taken into consideration together. And there are very important traits or ways to study ecology and evolution. For example, ecology mainly takes a focus on the spatial dimension of biodiversity, while evolution takes a focus on what is the temporal dimension of biodiversity processes. So, for example, while ecology might measure biologic or biodiversity changes in an order of few generations, sometimes evolution might take this change or might count this change in a term of sometimes millions of years. So, among other differences, another one that is important for our discussion today is that ecology mostly focus on the phenotypes of a species, that is mainly those characters that we can see, while in evolution the focus is on the relation between these phenotypes and the genotypes of these phenotypes. And so when we are talking here about management of wildlife, I think it is fair to say that we mostly take into account historically what is the information that comes from the ecological part, so from the processes that we can see in real time, that the ones that we have quantified. But there is a very important part of biodiversity, that a portion of this biodiversity that it comes from the evolutionary history of this biodiversity, that is mainly encoded in the form of genetic diversity. And this part is not always taken into account in the management of wildlife, maybe because we don't have this information, and sometimes we have it, but we don't know how to use it. So I guess that many of us will agree that there is a gap, there is a disconnection between academic research and wildlife management. I think this is general in all the fields of biology, but it's probably more abrupt in the field of conservation genetics. And that's probably because applied conservation genetics is quite a recent field, even if there is a lot of theory and knowledge out there that it's known for many years, but its applicability into the real cases, scenarios, is not always fully implemented. So there are lots of different potential applications that we can do from genetic data. This is just a broad scheme where you can see some of this, the scale of some of these applications. And this can go from genetic monitoring to gene editing to facilitate adaptation of some populations, and it can even go to cloning extinct species. However, I think that what many of us in this room might have in mind pretty much restrict to this area here, which is mostly what we could do, or what mostly thinking about conservation in endangered populations of butterflies. And in this case, you're working with Apollo. You have a species that is not extinct, that there are some populations that maybe are, is a species that is not a genetic model species, meaning that we don't have a lot of information about it. And it's also a species that have a very fast generation time, especially if we compare with large mammals, trees, etc. And those factors are important to consider. So the main problem when we need to, or we are engaging into conservation programs, is because we have small populations. Small populations can be the result of a multiple set of factors that Pavel mentioned, some of them, that can be measured and quantified from the ecology side of research, which is overexploitation, the fragmentation of habitats, habitat loss, the detrimental interactions with alien species, for example. All these are factors that I'm sure are going to be discussed very widely by many experts in this room. But all these factors usually drive our populations to be small enough in a way that they enter into what we call an extinction vortex, which is a suite or a process where a population starts losing genetic diversity and fitness because of several population genetics mechanisms like inbreeding depression, genetic drift, genetic load, etc. And the problem with this extinction vortex is an effect that it's very hard to recover. So once we are here, we need to be aware that we are already late. So once we have classified our species or populations as endangered species, it's because we're late. So the best way to avoid an extinction vortex is to prevent populations to decline. But okay, we are here, and this is what we have. But just as a note of caution, I would like to say that not all extinction events have to come from very small endangered populations. So there are cases, as for example, these two species that once were some of the most abundant species in North America, suffered a rapid decline and eventually got extinct in a matter of very few years. So what I want to say here is that we need to be aware that there are no species out there that do not need protection or conservation measures, and that once we are in the situation of small populations, what I was saying, it's probably because we didn't monitor that or we didn't see that coming, but we are there. So this might be the case of Apollo here. But okay, we have small populations and we need to solve that. So what can we do with genetic data? What can genetic data tell us? So we typically use as a main unit of work the genetic diversity. Genetic diversity is that biological variation or that set of genetic traits that are shared between any group of organisms. And optimally, we know that high genetic diversity, that is meaning like a large gene pool, is probably going to positively affect species resiliences over time. So ideally, we want to have populations that are large enough as to preserve or to maintain the levels of genetic diversity that are optimal for that population to survive. Genetic data can also tell us about genetic connectivity. What are the levels or directions of gene flow? Meaning that how the different populations are connected between them and to what extent there are some populations that have higher dispersal rates and are contributing to other populations in some different ways. At the same time, we can also learn what are the populations that are more isolated from each other, and consequently try to prioritize conservation actions towards some of these populations according to the connectivity criteria. There might be other criteria too. Genetic data also tell us about demographic history. And what is that important? Because demographic history tells us from where, in terms of population size, we are coming. So we can learn if there has been any historical decline of these populations that we just couldn't quantify, that it occurred before we can started monitoring. So that means historical declines is something that occur in evolutionary time, not in ecological time. And that's important because we need to know from where we're coming, or if there has been other pressures that we ignored just for observing these butterflies. Another thing we can measure with genetic data is effective population size. Effective population size is the number of individuals that one population needs to keep the genetic diversity that they have. And please note that this is different from the census population size. Census population size is the one that we can actually monitor thanks to our field monitoring experiences, and that it's close to the real number of specimens that we have in a population. Is different because census population size is typically higher than effective population size, meaning that there is a threshold for which any additional individuals that we include in a population, they are just not contributing much necessarily to new genetic diversity to that population. And those are important things to consider when we have talking about populations, about traits for conservation programs. I skipped that, which is fine. And I also like to talk a little bit about what are the different types of genetic variation that we can quantify. Because we typically say, okay, genetic diversity. Genetic diversity has a lot of different types of scales and scales as well. So, for example, we can distinguish between neutral and adaptive genetic variation. Neutral variation results from those stochastic processes that actually don't have any direct effect into individual fitnesses. However, adaptive variation would refer to that one that results from natural selection processes and that actually have a direct effect on individual fitnesses. Another types of genetic variation that we can look is the phylogenetic scale, where we try to understand what is the connection or the link between all these observed genetic diversity between different lineages. And then we have another kind of genetic variation that might be interesting, which is the interactions one. So we don't really focus on the genetic diversity of our target species, but maybe we should be focusing also on the genetic diversity of all those organisms that our species is interacting with. For example, endosymbiont bacteria, viruses, mutualists that are important for that species, other insects maybe, and why not host plants? So monitoring genetic diversity of the host plants of our target species might be an important action in conservation, because ultimately the host plants, what it happens to the host plants is directly affecting the butterflies. I would also like to briefly clarify some terms that we are also using when talking about genetic approaches. We typically use the term genetics when we focus on particular genetic features, like it could be the evolution of one particular gene or one portion of a chromosome. But in recent times, the term genetics has broadly expanded into genomics, which is actually thanks to the advantage that next generation sequencing techniques give us, we can actually look at the whole chromosome scale, the whole genome scale. So if we translate this into phylogenetics, we would be talking about phylogenomics, which would be using whole genomes to understand phylogenetic relationships. And lastly, the term metagenomics, it's also might be also interesting in conservation, because this is looking at also massive data of genetic diversity from the interacting communities in our habitat or for interacting with our target species. So all these are important terms to consider too. So now let's talk a little bit more about what is differences between neutral and adaptive variation in conservation. We mentioned that neutral diversity can give us very good information about indexes that we need for conservation. We mentioned demographic processes, we mentioned population structure, inbreeding coefficients, migration rates of connectivity. But what neutral variation don't tell us is if there are any particular selective processes that are going on out there that might be favoring or hindering any particular genotypes in a particular environment. And that's what adaptive variation can tell us. So there is an open debate about what should we be using in conservation programs, whether should we measure whole genome neutral variation, or should we focus on adaptive alleles that we know work well in one particular environment. This is an open debate, and it is an interesting debate. But probably anyone or most people agree that both things are not mutually exclusive, so they can work together, and that ultimately I think the consensus for now is that for natural populations, it is probably better to focus at the beginning, at least at the first place, in introducing general over whole genome neutral variation in the populations before focusing on adaptive diversity. But probably in the future, in the near future, or this is happening already, there will be more and more research on how to include adaptive variation into genetic programs. That is because at this scale we can, for example, learn if how many harmful mutations might accumulate one population, which is what we call genetic load, or for example, to map in a chromosome if there is the presence of any lethal gene that, of course, we don't want to introduce animals with this lethal alleles into a reintroduction program. Another scale that genomics give us in terms of understanding genetic diversity, much beyond the typical measures that are single mutations at particular sites, are those structural elements that also are important in evolution. So evolution acts in the genome at different scales, and these scales, the structural scales, might involve like inversions in the chromosomes, mobile elements and non-coding elements that actually have a very important function in regulating the genome to a point that this regulatory elements helps populations or species to adapt, the potential adaptive potential increases. So it is important also to get into account this kind of genomic features that are much beyond of typical classic ways to look at genetic diversity. So let's talk a little bit about genetic rescue. Genetic rescue is this artificial action where we add genetically unique individuals in a population with the goal to increase their genetic diversity and ultimately to make this population grow. And this is a very typical practice in conservation genetics. However, if we are going to do genetic rescue, we really need to know what we are doing, why we are doing, and what is this scenario. Because we might be interested in increasing the level of gene flow between populations because they are too disconnected. And generally, this seems to be like we're going to expect like a positive outcome. However, it might happen that we might be losing the potential of some. We might be rapidly using highly adapted alleles because of this interbreeding. So preventing population admixture in some cases might be also positive, even if we have small populations, because we might help those alleles, highly adaptive alleles, to stay, even in detriment of losing overall genetic diversity in the rest of the genome. So what I think it's important to say here is that there is no one single way to do or to act in genetic rescue or reintroductions, but that all of them depend on every single scenario that we need to do these assessments. So the more we know about the genomes, the more we can choose well our actions and can less likely to fail. So all the information at the genomic level is very important because of that reason. So we all probably want to see this kind of curves here that happens from reintroduction programs. Probably one of the most charismatic cases of reintroduction was with the Macrolinia butterfly in the UK, where short story is we had a decline, a rapid decline and extinction of a population. So we do a reintroduction, we introduce new specimens from far distant, non-threatened populations, and the population recovers, and we do magics, and everybody is happy. But this is not always the way. This sometimes might happen, some other times might not happen. There are many cases of failures as well. So I'd like to contrast this with the case, for example, of this is a famous book case, like about the genetic rescue of the Ice Royale wolves in North America, where the introduction of a single individual actually get down all the population, made the population collapse because it introduced non-adapted alleles into that population that were dominant alleles, and just the population ended. So what I want to say here is that there are cases of successful and unsuccessful cases, but we cannot be blind of what we are doing, because this is what it might happen. So let me go a little bit into butterflies, and I'd like to present this study on Melanargia raceae that Vlading and colleagues did some time ago. This is a species that was extinct in Hungary a century ago, and there is the interest to reintroduce it in Hungary. Okay, so the researchers made put this as an example because they look at different types and scales of genetic diversity in order to give the most the best possible advice in order to do these reintroductions. So what they did, something they did was they managed to sequence DNA from the extinct populations in Hungary, thanks to specimens that were stored in museum collections. And with this DNA material, so they built a phylogenetic tree so they could learn what were the closest lineages in terms of relatedness, and also they could measure what were the dominant genetic makeup of that species and of that population, and what other populations were more similar to them, with the goal to inform what could be the best possible sources. So here we have been talking about genetic structure, and you can see in this map by the different colors how genetic structure looks in this particular species, and we can see that there are mainly 3 different groups by 3 different colors in here. That means that these 3 populations have low levels of gene flow, so they are pretty characteristic between them. That's important in order to select the source. But also what the researchers did was to look at the endosymbiont strains of one particular bacteria, which is Wolbachia, which is a bacteria it's common in Lepidoptera, and it is known to have some effect on the fitness. So what they did was to sequence or to manage to know if the different populations have infected by this bacteria and by what strains. So with this they could inform also additional layers of information about the introduction of this species. They also look at the climatic similarity of the different populations to inform about the niche. So also again about the adaptive potential of any possible reintroduction. And with all this data at hand, they managed to conclude that the populations from from the Balkans and from East Ukraine were probably the best source to reintroduce the butterflies in Hungary. So now I drive you about on theory and some examples. Let's talk a little bit about Parnasius Apollo. What I wanted to say is that it is important when we are talking about one particular species to know from where the species is coming from. What is the evolutionary history of this species? And in the case of subfamily Parnassinae, it is quite interesting to see that most of the genera in the subfamily have pretty relatively restricted distributions and low number of species per genera, except the genus Parnasius, which shows a lot more and a lot faster diversification rate than their other subgenera. To the point that in about, don't remember about 16 million years, I think, they got up to 61 species out there. And this is pretty exceptional, pretty special in a phylogenetic sense. So, within the genus Parnasius, there's also interesting phylogenetic traits to observe. For example, we observe that in the subgenus Parnasius, here where the Apollo species belong, there has been, they use a different, or one host plant that is not used in the rest. So there is an important or significant evolutionary shift to take into account. But also what we observe here is that the subgenus Parnasius also show a higher rate of diversification than their relatives here, to the point that in 6 million years they have produced about 18 species. So this is also pretty exceptional, and denotes that this lineage here is experienced for some reason, some kind of higher diversification rate than their very genetically similar species in the other lineage. And all these are points that are important to take into account. So at the level of population genetics, and Pavel also showed this image, we have learned, there have been some studies there, we have learned about the population range, colonization history, we have learned about some demographic patterns, about what are the populations with higher effective population size, lower effective population size, what has been the reasons of these fluctuations over time. And most recently, we have also gone a little bit deep into the phylogeography in a comparative way with other species in Europe. And this is for Parnasius Apollo, and we have learned that there's mainly three genetic groups. The genetic structure is divided in mainly three genetic groups, and some of them are quite genetically diverse. And if we look at this diversity in a comparative way with the rest of the species in Europe, all the species, this is what we can extract from this plot. So we have species that have a lot of genetic variation, but little structure. This would make sense, for example, for species that have high dispersal rates, for example. But then we have species that have very few genetic variation, but have a lot of spatial structure. That would correspond, for example, for very restricted endemics. In our case in Apollo, we are in this area here, which means we have a lot of genetic variation, and we have also a lot of genetic structure. So what does it mean? That means that there is genetic diversity out there for Parnasius Apollo, but that means that this is very structured in different populations. And that tells, okay, there is genetic power there for reintroductions, for conservation actions, but we need to be very aware what we are taking for these reintroductions, because this could be very different populations and lead to outbreeding depression or lead to non-adapted alleles, and we would just do useless efforts in conservation. So this is pretty much what, in very general scale, what I think it is known about European Apollo in terms of population genetics. But there is an important newer contribution, which has been published for the first time, the genome assembly for this butterfly, and this is a very good thing because it opens the door to go much beyond what we are doing now. So we can, for example, start looking at those genes that have adaptive potential in a deeper way. So this is very good news, and one very interesting finding when sequencing this genome was that it turns that it's very big. It turns to be one of the biggest known genome among butterflies and moths. And this is quite interesting. Maybe it is not that surprising, because if we look at the evolutionary history of this lineage, we have seen that it is quite special. And we know that the evolution of the genome, it's pretty often involved in diversification of lineages. But the reasons why this is this way, I don't know. I don't think anyone knows yet, but it's probably multiple reasons and multifactorial. But this is an interesting finding. The way why this genome is that big seems to be related to an increased number of DNA content in repeats, which is a kind of genomic trait that is hard which function is hard to determine and is unclear, but that are definitely known to have some important regulatory functions in some cases. So with this, I finished not before showing this slide that I think it's pretty interesting because it illustrates how in conservation genetics we take progressive steps according to what we keep learning from our genetic data, because we don't know everything at once. So you can see that here different information is classified in high or low in terms of genetic data. Who and how this is classified or the threshold is established as high or low depends on every single study and every case scenario. So take-home messages. Main problems come with populations are small. We know that, but we need to work on prevention to avoid this happening, because preventing the populations to decline is much better than to cure. This prevention requires monitoring, phenotypic monitoring, demographic monitoring, but also genetic monitoring. These genetic assessments, either at the phase of monitoring or the phase of genetic rescue or introductions, are very important for conservation policies, but at the same time are expensive, requires expertise, etc. So we need to drive well our efforts. Also get with the idea that there are different types of and scales of genetic data to look at, not only one. And finally, as I said several times, conservation actions require case-by-case assessments. There's no universal protocols, and that's why we are here discussing about the conservation of one particular species. So with this, I finish, and I don't know if there are time for questions or discussion. Thank you.
Thank you very much. It was really very interesting presentation, at least for me. That was very valuable input for the project. Are there any questions? Okay.
Question?
Hello.
Can you hear me? Oh, all right. Hear me? Oh. What is the password to it? My name is Dušan Šacha. I am from Slovakia, a state nature conservancy, and what interested me was your example of that wolf, Isle Royal wolf, and that allele that was recessive but gave advantage to the population. I don't know that example, and can you tell me something more about it, because it seems counterintuitive?
what I know about that example is that they introduce, without knowing, one allele that became dominant but was not adaptive to that. Sorry? I don't know. I don't know.
I would like to add one comment. You told about local adaptation, but I would like to mention that in some specific cases, local adaptation may be also endangered from population. This local population, because it is mainly the kind of dispersal potential. There are several data suggesting that in small population, migratory activity is selected out because it means that individuals which migrate from subpopulation died. However, from the conservation point of view, it means that population collapse and make smaller and smaller. There are few. I know the two examples of such. One is the swallowtail in Great Britain part, and the second one was Parnassus apollo in Pieniny. Due to long-term isolation, individuals became extremely sedentary. In fact, it was adaptive, however, very dangerous for this local population. It's the problem of, adaptive landscapes. Thank you.
Well, yes, I agree that that needs specific assessment. It's a comment, but as I said, this is the use of adaptive variation in conservation genetics is an open debate, and this is why the reason. No? There are cases and cases.
It's not unique way.
Mm-hmm.
I do have one question. Max from Austria. I'm just going to speak loud. The question is, we are encountering very often.
Keep going.
We are encountering very often a position from authorities that basically suggest or would like us to, if we talk about supporting a remnant non-survivable population or reintroducing a species, that we only use the species that has been there or that was there many, many years ago. When I looked at your presentation, I mean, there's no standard answer. It's very clear. But you seem to hint that the genetic variations are also should be looked at, so that we actually do not only introduce the one that was there like again and again and again, but actually open our mind to say there could be subspecies or a different one from this or there, so that we basically help the species to do what it cannot do right at the moment, travel these long distances to be able to adapt. Is this correct? Did I understand this correct?
Yes, I would say yes, and I'd like to comment on this. I think that at the moment that we are reintroducing a population, we need to be aware that the lineage or the population is extinct. So the genetic diversity that was accumulated in that population is lost. That's for sure. We're not going to recover that ever else. So in the moment that we are introducing one population or refilling one population with another one, we're cloning that population in another place. So why is this important? Needs to be assessed in a global scale, in a connectivity scale. We want to refill that place because that place is going to help the other populations to decline, but not necessarily just because it was there. We need to put it there, unless we are looking at the ecosystem services that is providing. That's another story. That's I don't know if that's my comment.
The story about improving migration potential is the story of Polish and Slovakian Parnassus Apollo Frankenberger in Pieniny. Because when we start reintroduction project, we have the great problem because individuals were extremely sedentary. Female didn't want to fly just because of probably the several decades of natural selection against migration. However, in the Slovakia part of Pieniny, there is bigger population of the same subspecies. And we found, and they are very active, migrating. And we, in 1996, Bogdan, it was in 1996, we have decided to introduce the Slovak butterfly in our population. And effects was really good. In the first year, butterflies start to migrate, and the most of migrators became from the Slovak of mixing Polish-Slovak line. But it was very risky because we are afraid that we are afraid of the outbreak effect. However, there were data that in 20s and 30s, these two populations were connected. So it was why we decided to mix them, and the migration amount increased. Now the longest migration is more than 10 kilometers by one individual. Thank you.
Thank you very much. Are there any questions? Okay.
Yes, Jorge Baixeras from Spain. Yeah, I was interested in your talk. And I was curious about the genomic size of the Parnassus. You mentioned the genomic size, but how much of this size is just junk? I mean, the genomic size doesn't mean anything, especially the size. I mean, it may be usable or not. What do you think?
It is fourfold bigger than average on the rest of butterflies, more or less. So that's quite a lot. It's not, doesn't seem to be some kind of chromosome duplications, etc. So it seems that mostly relates to repetitive content that we know its functions are uncertain, if any functions. So as you say, probably a lot of this is junk. And to some extent, it's not known what repetitive content is very hard to determine, and it's not known. But I think that what it denotes, at least, that there's something going on in that lineage at the genome level, that it's fostering evolution at the genome level. So, yeah, how much is, as far as I read, I know as you know about the genome. It seems to be repetitive content. The authors of that work mostly says it's repetitive content. That's what we know.
Okay, thank you very much. Still questions? Okay, thank you. Thank you very much. And I would like to introduce our breeders in the project from 3 countries. So from Austria and Czech Republic and Poland. And Max, you were chosen to start. Our colleague from the EWS from Austria will introduce our breeding techniques and what we do in the project in these terms.
Actually, I'm Otto. No, just joking. Otto is our breeder, but he had a personal medical emergency, so he could not attend. So he says his kind regards, which actually means, Magdalena, that we will come back with him in a couple of weeks because he has 35 years of experience. And I'm hoping I can go through the slides and explain to him. If not, I will call back to my colleague Magdalena, but maybe Magdalena, you can come up and help me out if I don't really get everything correctly. So Otto is now retired. He has 35 years of experience. And in Austria, we have a declining Apollo population. It is very fragmented, and it's not being helped by the bureaucracy of the nature conservation organizations. The authorities are basically blocking everything that surrounds these reintroductions or supporting existing populations, which is making it very difficult. And the argument and the reason is very simple. They are afraid that if we introduce an Apollo, it will become a Natura 2000 site. And the moment you have that, you have the agricultural community against you, so they just are shying away from it, even though everybody knows it's urgently needed. Otto's experience has been that basically he does everything by hand. It's very personal, the way that he breeds these butterflies, these Apollos, and he has many different subspecies that he does that, and he is using it basically in his backyard. He's got plastic containers, and that's where he does it. Now, in this project, we will elevate and professionalize his breeding, but it shows that even without professional systems, for 35 years, this species allows you to basically do homegrown breeding. Now, this is not a call for anybody to do that, to copy it, but he has had huge experience. This slide, I have no idea what this is about, but it basically is a development cycle. I'm responsible for the citizen science part and the marketing. And it is a language I do not understand. Maybe somebody can help me out?
Yes, there are the months, January, February, March.
Right.
There are particular stages.
I got it. So these are the 12 months, and this is basically the life cycle of the Apollo as it is going through the annual cycle. Hey, I got it. He basically is using these plastic containers. The eggs are laid on the host plants and directly next to them. In June and July, 10 to 14 days, the caterpillars develop. The hatching then happens in February and March. The eggs are hibernated in these small plastic boxes. His are not always that nice. They are much more simple. The humidity is very important, and obviously continuously checking if everything works out. There you see his breeding center. And I'm telling you, it is literally the backyard. So this is what he does, and he has been doing it for 35 years, highly successful. And what he does, depending on the temperature, now this is already in the presentation today, we have in Austria rapidly climbing temperatures. When we talk about climate change plus 2 degrees, we have plus 4 degrees. We have plus 6 degrees. And the weather is becoming very unstable. We already had snow, which normally in September happens short, but now already we have a complete week. So the impact on the whole ecosystem, including the insects, is a big unknown for us. Avoid too intense sunlight, alternate, fill the boxes, and this is primarily with the rocks and stones so that they can, if the sun is too strong and the amount of sun we are getting is increasing by 10% over the last couple of years, that they can actually go into the shade. Then you move the caterpillars to larger buckets. Don't put too many of them in there. Be careful, be clean with, like I said, any kind of breeding effort, that you do not introduce any kind of other diseases into them. And then after 4 to 6 weeks, they start to run hectically, so they are kind of in an emergency mode to find a place where they can go into pupation. So he then creates hiding spaces for them. So using branches and again, different kind of things that the caterpillars can hide themselves. And again, we know, as we heard in the previous, they hide from the ants. They hide from the predators. That's what they do. Now, we don't have any ants in the boxes, so they would not need to, but their gene system tells them, I need to hide. So you have to give them something to hide. After two to 3 weeks, then at sunrise, and it's precisely at sunrise, they start to then leave the pupae. And there you have to be very careful because they then need to, as we heard earlier, spread their wings. And if they then get disturbed, those wings are damaged and basically you lose that butterfly. And then life begins again from new. These are the sedums that are specialized on. Now, we are already there running into problems because of climate change. Just so you're aware, our tree line is rising by 300 meters. Especially in the high alpine meadows, we have temperature rises that are incredible. And with that, it's also the plants that normally the Apollo needs are changing. The Rhodolia roseo and Sempervivium montanum are the ones that our Austrians ones at least, like the best. If you go to Italy, his experience has been that these are the sedums that they have There we go. And he has been experimenting also with other plants, and that has been his experience that he would like to know, but this is not scientifically it's just his personal observation. But I think that there's much more research we need to figure on, because I think there will also be, because of climate change, a change in the plants that we are faced on. This is the challenge we are running into. We have the sites, we know which sites we have. We have run into what I said before, that the authorities insist this specific subspecies, it's not in there, you need to release exactly that subspecies. And what the hell do you do? So we are looking for support in this project that we can be much more freer in which then to release or support an existing population. The other observation, I'm in a different life project with shepherding against the wolf, no joke. And we have now discovered the Apollo at 2,600 meters in rocky alpine places. So what we are observing is that the Apollo is climbing up the mountains very fast, and we are thinking about actually starting a little citizen science project to get shepherds to send us photos, because I think we are overlooking Apollo populations in areas where the typical butterfly specialist does not go. Because when you look at those areas, this is high alpine rock glacier areas, and they are only concerned about hiking up to the peak and back down. They don't look at butterflies. But we have shepherds up there now, because otherwise the wolf would kill all the sheep. And we are thinking about maybe using them a little bit to identify remnants population or new populations that we just do not have on our radar system right now. Yes. Was that quick enough?
Yes, thank you.
Thank you. Any questions?
I think at the end, so we will have all the presentations. This would be a little bit different. Milos, yeah? So now our Czech partner. Milos, you will be presenting in Czech, yes? Okay, so Milos will be presenting in Czech, so those who do not understand Czech, please use your headphones.
Aha, on tam ještě neskočil. No. Počkáme, až nám to naběhne. Mhm. Proč to neběží? Nechápu. Takže dobrý den, jmenuji se Miloš Andrés. Chovem motýlů se zabývám už od dětství. Nejprve jsem to dělal všechno víceméně výzkumně pro sebe. Zajímalo mě, jak ty motýly fungují, zejména mě zajímaly potravní preference u některých druhů, protože ne všechny druhy v literatuře jsou zmíněny přesně tak, jak je to tam popsáno. Spoustu věcí tam je ještě nedořešeno v tomhletom ohledu. Takže mě zajímaly ty potravní preference, dále mě zajímal takový ten skrytý způsob života larev. Zpočátku se to dalo dělat v nějakých lehkých zařízeních typu nějaký zavařovací sklenice nebo nějakého akvária. Později jsem na to musel začít vybudovávat různý druhy zařízení. Jak vidíte na tabuli, tak je tam de facto zařízení typu foliového skleníku, ale vlastně to není potažený folií, je to potažený takovou tou plastovou sítí na lešení, která je světlostabilní, poměrně pevná a ty chovy poměrně dobře chrání, je taky prodyšná. Vevnitř je tedy, nebo nahoře je tedy srážkový stín, každý to zařízení má střížku, musí se nějakým způsobem vlhkost regulovat, zejména v zimním období, takže tu si regulují sám. Tady na těch obrázkách to ještě není, a na těch dalších, co budou, tak je taky zařízení právě z důvodu nadměrné vlhkosti postavený buď na nějakým ztraceným bednění nebo na podezdívce, která zase brání vstupu ty vodě spodem těmi drenážními otvory v těch květináčích. Zařízení je vysypaný klasickým křemičitým pískem a v něm jsou až po okraj zapuštěny ty jednotlivé květináče, v nich jsou nasázeny ty živné rostliny a zároveň potom, když jsou tam kladeny ty housenky, tak samozřejmě musí být kladeny v adekvátním počtu s ohledem na to, jaký druh motýla se chová. Jo? Všechno tady je zapuštěné až po okraj v tom písku. Každý ten květináč je individuálně přikrytý ještě jemnou monofilovou sítí, která brání vstupu různých parazitů, predátorů do toho prostoru a nějakým způsobem nemůžou potom ovlivnit vývoj toho chovu. Ten materiál je natolik dobrý, že i když ty housenky lezou po stěnách vlastně těch jemných monofilových zařízení, takže nedochází ani takhle nedochází vlastně k nějakému parazitaci nebo nějakému ataku těch parazitů na ty housenky. Podobným způsobem tedy, jako chovám jiné druhy motýlů, jsem tedy začal letos chovat i toho jasoně červenokého. Chovám to vlastně dvěma postupy. Jedním tím osvědčeným postupem chovám po vzoru polských kolegů, které teda v těch plastových dózách s nějakým suchým substrátem, dává se jim řezaná potrava. Nějakým způsobem se vždycky housenky musí zase držet v nějakým adekvátním množství, to už tady taky bylo zmíněno, aby nedošlo k nějakému napadení zase s nějakýma houbovými chorobama nebo něčím takovýmhle. Takže ty plastové dózy jsou potom dělané tak, že mají to plastové víko, to je proříznutý a je tam opět místo toho vložená monofilová tkanina, která zase brání vstupu nějakých predátorů a parazitů. Druhým způsobem, protože jsem byl zvídavý a zajímalo mě, jak vlastně Jo, vidíš to. Já jsem zapomněl, co? Jo, počkej. No to jsou ty plastové dózy. Já jsem vlastně zapomněl zmínit, že tady v tom mým zařízení prošlo spoustu druhů denních motýlů. De facto od roku 2010 se zabývám nejenom výzkumnými chovy, ale také těmi reprodukčními chovy. Je mezi tím trošičku rozdíl, protože při tom výzkumném chovu není potřeba držet takové množství jedinců, jako právě při tom reprodukčním chovu. Takže tam byly velké změny a vlastně od toho roku 2014 už jsem zapojený v nějakých takových těch záchranných programech pro ty jednotlivé denní motýly, které jsou tady po různu zmíněny. Tady mám nějaké tuchovné zařízení. Jinak tedy toho jasoně červenokého jsem ještě zapomněl zmínit, tady dělám podobně jako ty motýly polopřirozeným způsobem, a to v těch větších plastových květináčích. Protože jsem byl zvědavý na tu potravní preferenci, tak jsem si vlastně toho pěninského Apolla, který je prej výrazně závislý na jedné živné rostlině, a to je to Hylotelefium maximum, tak jsem nasadil tedy květináče s tímhle maximem, pak jsem nasadil čistý květináče s rozchodníkem bílým a také jsem udělal kombinaci obou dvou rostlin, protože mě zajímalo, jak vlastně ta potravní preference bude vypadat, jestli budou preferovat jenom jednu rostlinu nebo se pustí do obou dvou. Takže to bylo velmi zajímavé, zejména z toho důvodu, že v těch plastových dozách, když jsem pak rovnal velikost, tak se v těch plastových dozách ty housenky vyvíjely na tom maximum víceméně vyrovnaně, kdežto v těch plastových květináčích na těch různých druzích rostlin, a to i na tom Telefiu maximum, tak ty rostliny byly velikostně velmi rozdílné. Byly tam některé velmi malé a některé už třeba 2 cm. Jinak teda já jsem to potom dělal, takže jsem svým způsobem nechal ty housenky do toho v těch květináčích do stádia asi 2 cm a pak jsem je také vrátil do plastových doz. Držel jsem je tedy odděleně od těch housenek, které byly od začátku drženy v plastových dozách, právě protože mě zajímalo to, jestli ty housenky nějakou ty menší doženou tu velikost, nebo co se s tím bude dít ve finále. Takže jsem to držel odděleně. Pak jsem jim vlastně podával taky už tu řezanou stravu. Přešel jsem čistě už jenom na to Telefium maximum, protože jsem zjistil, že opravdu jiný druhy potravy, kterou jsem jim dával skrze ty rozchodníky, tak nebyla tak pro ně asi adekvátní, protože ty housenky opravdu zaostávaly v růstu, nějakým způsobem začaly i vypadávat z toho začaly hnout, takhle to řeknu, začaly hnout postupně. Takže jsem je potom držel výhradně na tom Hypertelefium, nebo na tom Hylotelefium. Vlastně v těch dózách byly až do toho stádia toho kuklení. Svým způsobem jsem jim do toho rohu těch zařízení postupně nainstaloval papírové kapesníky anebo ubrousky, které sloužily jednak k tomu, že ty housenky se do toho rádi přes noc schovávaly a pak se tam v zápětí začaly kuklit. Kukly jsem průběžně odebíral, dával jsem je do takových těch středně velkých monofilových houseníků. Tam de facto byly už do toho závěru. Aha. Jo.
Neříkej toho tolik, on to nezvládej překládat.
Jo. Já moc milu.
Zabíjeli těch podrobností a zkus taky ty český názvy, takový květináč a tak, oni to neznají, jako napodobovat.
Jo, takhle. Jo. No, jasný. Dobře. Nejsem moc běhlej v těch diskuzích, takže se omlouvám, třeba o nějaký prohřešky drobný. Takže ty kukly potom byly průběžně dávány do těch středně velkých monofilových houseníků. Tam pak probíhalo i to líhnutí těch motýlů, odtamtud jsem ty motýly odebíral a dával jsem je do větších monofilových houseníků, zvlášť jsem si dával samičky, zvlášť jsem si dával samce. Krmení probíhalo výhradně v ruce, to tam možná taky někde už bylo vidět. Probíhalo v ruce, to, že jsem motýla držel, nalil jsem mu vlastně do víčka od PET lahve medocukrový roztok s příměsí nějakých vitamínů a potom jsem vlastně těm motýlům nabídnul tu potravu, oni začali sát. Že jo. Jinak páření pak probíhalo v takových větších proletových zařízeních. Zároveň jsem potom zkoušel páření i v takových těch menších monofilových housenících. Všude jsem tak nějako zaznamenal postupně za tu dobu nějakou kopulaci, ale nebyl jsem s tím plně spokojený, protože některé ty samičky, které nebyly oplozeny, tak třeba začaly, během 4-5 dnů klást neoplozená vajíčka právě na stěny toho zařízení. Takže jsem potom přistoupil k umělé vlastně kopulaci, že jsem motýly pářil v ruce. Tuto techniku znám i vlastně, nebo takhle běžně ji používám i u jiných druhů, který chovám. Většinou to jsou druhy, který nechtějí v tomto ohledu moc dobře spolupracovat a zároveň jsou to třeba i motýly, u kterých si vedu nějakou přímou plemenitbu nebo něco takového. Jo. Takže jsem si tohle osvojil. Dalo se to dělat poměrně potom dobře, že jsem třeba měl 25 samic a dalo se během 2 hodin bez problému chytit těch 25 párů. Jo. Kladení vajíček pak probíhalo trošičku jinak, než mě vzdělili polští kolegové, kteří to mají v takových těch monofilových sáčcích. Já jsem to nechával vyklást v klasické plastové dóze. Byla to spíš nižší dóza, zase proříznutý víko, ale to bylo nahrazeny ne tylem, nebo ne monofilovou tkaninou, ale dával jsem tam tylovou tkaninu. A samičky tedy kladly, ještě jsem zapomněl zmínit, že jsem tam dával čistý křemičitý písek, ten byl mírně vlhký, aby se zachovalo lehce mikroklima, takový vlhký, aby se ty samice zbytečně nevysušovaly. A vždy pečlivě nakrmený jsem je tam vkládal. Když byla teplota kolem těch 25 stupňů a ke stimulaci kladení jsem jim tam přikládal nějaký nařezaný kousky rozchodníků bílých, načež potom bez problému kladly vajíčka, které jsem potom k večeru odebíral. Musel jsem přitom hlídat velmi teplotu, to už tady taky bylo zmíněno, že to je druh, který je poměrně háklivý na tu teplotu, takže jsem musel nějakým způsobem hlídat ty hodiny, které jsem nechal ty samice aktivovat v tom kladení. Obyčejně to bylo, že když to bylo do těch 25 stupňů, tak jsem je dával vždycky kolem té 1 hodiny odpolední, když bylo sluníčko, a kolem nějaký 5. 6. hodiny jsem je odebíral. Většinou už v té době prokazovali takovou, jako nebyli už aktivní, spíš byli pasivní, seděli se složenými křídly. Tak jsem je odebral, i hned jsem je nakrmil. Když byla teplota kolem těch 30 stupňů až nad 30 stupňů Celsia, tak jsem obyčejně samičky nechával klást pouze v dopoledních hodinách, třeba jenom na 2 hodiny, a nebo jenom odpoledne zase k večeru, mezi nějakou 5. 6. hodinou, protože když jsem je vystavil nějakým těm přímým teplotám kolem té 12. nebo 1 hodiny, tak mě ty samičky kolabovaly. Jo, zpočátku začaly klást a potom kolabovaly. Jo. Tímhletím postupem se mi podařilo získat asi těch 4000 vajíček vlastně pro následující sezónu a pro to otočení toho chovu. Tak to je tak asi všecko, co jsem chtěl říct.
Thank you very much. Dziękuję, Miloš. Roman? Our new team member, Roman Manak from Czech Republic.
Yes, thank you. Hi all, I'm Roman Maňák from Czech Republic. I speak in Czech. Jsem ze vzdělávacího informačního střediska Bílé Karpaty, což je jeden ze 6 projektových partnerů. Bílé Karpaty je pohoří na pomezí České a Slovenské republiky a je to jedna z funkčních projektových oblastí. V české části Bílých Karpat vyhynul jasoň červenooký Parnasius apollo před několika desítkami let a v současné době stojíme úplně na samotném začátku projektu a se samotným chovem motýla ještě nemáme úplně zkušenosti, tak jako kolegové z Rakouska, z EVS, z Polska, případně z ČSOP Hradec Králové. Takže se omlouvám, že vám nesdělím naše zkušenosti z chovu, který u nás teprve začne v příštím roce, ale dovolte mi, abych se s vámi podělil aspoň o to, jak jsme se v letošním roce připravovali na to, jak nám vypukne v dalších letech chov tohoto krásného motýla. Takže samotnou chovnou stanici, která by měla vyrůst v nejbližších měsících a měla by vypadat zhruba tak, jako na obrázku vlevo, tak samotnou chovnou stanici jsme se rozhodli umístit v malé obci v podhůří Bílých Karpat, v obci Blatnička, a domluvili jsme se soukromým ekologickým zemědělcem, že tady tato farma vyroste na jeho pozemku, jehož obrázek vidíte vpravo. Samotné chovné zařízení bude vypadat tak, jak vidíte na obrázku vlevo. Uvnitř bude potom množící stan pro motýly. Mimo to se zde bude chovat nebo pěstovat živná rostlina pro housenky motýla. A živná rostlina se bude pěstovat i v těsné blízkosti tady toho množícího zařízení. Letos jsme se zaměřili především na samotnou živnou rostlinu, protože nemáme zajištěné nebo neměli jsme do letoška zajištěné pro další roky, kdy u nás ten chov motýla vypukne, neměli jsme zajištěné dostatečné množství živné rostliny, takže letos jsme zkoušeli pěstovat živnou rostlinu na 3 místech. Jedním z těchto míst byla střední zahradnická škola v Rajhradě, kde dokonce jeden student bude na chov motýla Apolla a na pěstování živné rostliny dělat závěrečnou práci. Soustředili jsme se na to, jaká je třeba úspěšnost nebo jaká je klíčivost semen, jestli se dají u tohoto druhu pořizovat s úspěchem třeba listové řízky nebo stonkové řízky a další aspekty pěstování této živné rostliny. Kromě toho jsme na vybraných lokalitách v Bílých Karpatech sbírali pro výsevy v blízkosti chovné stanici nektarodárné rostliny pro dospělce Apolla. Povedlo se nám nazbírat snad dostatečné množství, alespoň prozatím, 2 druhů chrb. Konkrétně jde o chrbu Centaurea jacea a Centaurea scabiosa. Omluvám se. Tak, kromě toho, protože máme pocit, že plochy, které máme vyčleněné pro pěstování toho rozchodníku jakožto živné rostliny pro housenky motýla nebudou dostatečné, tak jsme se domluvili se zástupci města Veselí nad Moravou, které leží nedaleko Bílých Karpat, zda bychom mohli využít některé jejich plochy, které jsou v současné době nevyužívané. Napadlo nás využít plochu, která asi před 30 lety sloužila ještě jako skládka komunálního odpadu. Yes. Rozhodli jsme se využít plochu, která ještě asi před 30 lety sloužila jako skládka komunálního odpadu a před 30 lety proběhla rozsáhlá revitalizace. Od té doby tady tato revitalizovaná skládka vlastně leží sadem a její svahy spontánně zarůstají vegetací. Pouze vrcholová část té rekultivované skládky je zatím zarostená nezapojenou vegetací a vzhledem ke klimatickým podmínkám a expozici skládky se nám jeví jako vhodné místo pro pěstování živné rostliny. V našem případě to bude převážně Hylotelephium maximum. Tak, děkuji za pozornost a doufám, že nám budete držet palce, že se nám po mnoha desetiletích podaří navrátit tohoto krásného motýla do Bílých Karpat, do české oblasti Bílých Karpat.
Thank you very much, Roman. Thank you. And now the representative from the Karkonosze National Park, Dariusz Kuś.
Dzień dobry Państwu. Moja prezentacja będzie w języku polskim. Nazywam się Dariusz Kuś, jestem pracownikiem działu ochrony przyrody. Zajmujemy się niepylakiem Apollo od 2016 roku. Hodowla niepylaka Apollo to jest w naszym przypadku gra zespołowa, bo oprócz niezbędnej infrastruktury do hodowli, którą zaczęliśmy budować w 2010 roku i przez 6 lat namnażać roślinę żywicielską dla tego motyla, to też bardzo ważnym składnikiem są ludzie, którzy tworzą tą hodowlę. Jest nas 10 osób, z czego 6 osób jest zatrudnionych z projektu oraz wspierają nas wolontariusze, którzy pomagają nam w działaniach, które prowadzimy w terenie, ponieważ staramy się, żeby nasz projekt nie zamykał się tylko w obszarze Parku Narodowego, ale wychodził szerzej, tak żeby finalnie spróbować wytworzyć metapopulację na tym regionie. Nasz materiał genetyczny do hodowli pochodzi z Pienińskiego Parku Narodowego. Jest to najbliższa lokalizacja, ponieważ w Karkonoszach ten gatunek około 100 lat temu wyginął i ten unikalny genotyp niestety już jest tylko dostępny do analiz, ale jako materiały muzealne. W Karkonoszach niepylaki startują dosyć wcześnie, bo to jest już luty. Natomiast co ciekawe, pojawiają się w momencie, kiedy nie ma rośliny jeszcze żywicielskiej. I w pierwszym roku było dla nas to bardzo frustrujące prowadzenie tej hodowli, bo mieliśmy wrażenie, że po prostu wszystko się to nie uda. Ale przy użyciu odpowiednich metod, a w tym przypadku bardzo istotne są suche kwiatostany rozchodnika, to ta hodowla się udaje. W pierwszym momencie, kiedy gąsienice są bardzo małe, one mają około 2 mm długości, tak więc jest bardzo ciężko je przykładać, żeby ich nie uszkodzić. Początkowo używaliśmy plastikowych patyczków do uszu, ale w tym roku nasza piękniejsza część naszego zespołu zwróciła uwagę, że lepsze są pędzelki do makijażu, tak więc nie można ich używać wielokrotnie. Tak jak Państwu mówiłem, na początku bardzo istotne są te suche kwiatostany, ponieważ u nas przez to, że nie ma dostępu, pojawiają się wcześniej niż roślina żywicielska. Gąsienice przez około 2-3 tygodnie tworzą takie lustra na tych suchych kwiatostanach. Pomaga im to regulować ciepłotę ciała, one są koloru czarnego, łatwiej się nagrzewają i w ten sposób spędzają początek hodowli. Tutaj podobnie jak koledzy i z Austrii i z Czech, te rozwiązania może się troszkę różnią wielkością, natomiast one wszystkie zmierzają do tego samego, czyli do ograniczenia czynników, które mogą spowodować dużą śmiertelność wśród gąsienic. I tutaj bardzo ważnym czynnikiem jest ograniczenie i niwelacja sytuacji związanych z warunkami pogodowymi, które mogą być w naszych warunkach akurat bardzo zmienne, bo epizody z opadami śniegu w maju w Karkonoszach nie są niczym zaskakującym, natomiast chodzi o to, żeby te gąsienice izolować od dużej wilgotności, ponieważ temperatura nie jest dla nich problemem. Te niskie temperatury tylko wpływają na tempo jak gdyby rozwoju, natomiast duża wilgotność, czy duże opady, czy obecność śniegu powoduje dużą śmiertelność. Te boksy również niwelują oddziaływanie wszystkich czynników oporu środowiska, a bynajmniej większości, czyli drapieżników. Tutaj to ograniczenie właśnie dostępu mrówek jest bardzo istotne, parazytoidów i w tym pierwszym etapie przebywają w takich małych boksach. W moment, kiedy pojawiają się żółte kropki na gąsienicach, jest to najfajniejsza część hodowli, bo wtedy już wiadomo, że wszystko idzie w dobrą stronę. Jest to pierwsza wylinka tych zmian garniturów, jest 5. W ostatnim stadium gąsienice są bardzo duże, czyli z 2 mm przechodzą na wielkość małego palca. Tak więc to już jest taka mała fabryka biomasy, która ma bardzo duże zapotrzebowanie na pokarm. W naszym przypadku, bo jest to pochodzenie pinińskie, my używamy głównie sedum maximum jako rośliny żywicielskiej. Czasami też jest używany sedum album albo Rhodiola rosace. Ta Rhodiola rosace wzięła się też stąd, że historyczne dane o niepylaku mówią, że występował w Karkonoszach w śnieżnych kotłach, a tam główną rośliną dla niego żywicielską byłaby właśnie Rhodiola rosace. Staramy się mieć bardzo dużo rośliny żywicielskiej, ponieważ w tym roku w końcu to zmierzyliśmy. zużyliśmy w trakcie sezonu 300 kg rozchodnika Sedum Maximum. Tak więc zapotrzebowanie na tą biomasę jest bardzo duże. Też jest bardzo ważne, że rośliny żywicielskie reagują pod wpływem żeru, dlatego najłatwiej to zauważyć na Sedum album, który ma bardzo silną reakcję chemiczną na żerowanie i praktycznie po kilku dniach widać, że gąsienice już przemieszczają się i szukają nowych roślin, ponieważ ta reakcja w roślinie była tak mocna, że po prostu opuszczają ją. Natomiast my wyszliśmy z założenia, żeby mieć jak gdyby 2 razy więcej tego rozchodnika na sezon, ponieważ używamy w danym roku 1 partii, a w następnym drugiej, tak żeby cały czas nie eksploatować tych samych osobników. No i większość pokarmu podajemy po prostu w postaci ciętego rozchodnika, tak żeby po prostu nie było reakcji rośliny. Natomiast przy tych ostatnich stadiach to wygląda w ten sposób, że to, co się wrzuci rano do hodowlarek, to po kilku godzinach już to znika. Jak gąsienice są w 4. i w 5. stadium, przenosimy je do większych hodowlarek. To jest bardzo istotne ze względu na warunki higieniczne w hodowli oraz na warunki związane z wilgotnością. Natomiast w przyszłym roku na pewno już mamy przygotowany 1 namiot, gdzie byśmy chcieli te gąsienice w starszych stadiach już trzymać na dużym obszarze, żeby miały większe jak gdyby pole manewru, ponieważ w tym roku mieliśmy duży problem z wysokimi temperaturami. Były 3 takie epizody, które spowodowały dosyć dużą śmiertelność i ona mogła być związana z tym, że jednak warunki w tych boksach są podbijane przez temperaturę. Przepoczwarczają się u nas gąsienice w boksach, my je później zbieramy. One zazwyczaj mają różne tam strategie, albo zakopują się w ziemi, albo gdzieś podwieszają w kątach boksów. I jak już mamy te poczwarki, to je przykładamy do specjalnych hodowlarek, z których później jest łatwiej wyciągać motyle. Poczwarki się pojawiają zazwyczaj w maju, końcówka maja i przez okres 2-3 tygodni są w tych hodowlarkach, no i później zaczynają się wylęgi. Wylęgi zawsze się zaczynają w ten sposób, że pierwsze pojawiają się samce, jest to naturalna właściwość tego gatunku. Po około tygodniu zaczynają się pojawiać samice i później te 2 fale samców jest coraz mniej, a pojawia się coraz więcej samic. Bardzo istotne w hodowli też jest odpowiednie wyciąganie tych wcześniej motyli, bo te skrzydła faktycznie są bardzo delikatne i jak jest ich za dużo w hodowlarce, to po prostu bardzo łatwo się uszkadzają, dlatego zawsze rano je wyciągamy i rozkładamy na tkaninie, tak żeby one mogły spokojnie napompować skrzydła. I tutaj też działa jak w szwajcarskim zegarku. Do południa tylko te wylęgi się odbywają, później już nie trzeba tam zaglądać, bo po prostu wszystko się kończy do połowy dnia. Po parowaniu motyli, które jeszcze tu wrócę, odbywa się w dużym białym namiocie. Kolor tkaniny jest bardzo istotny. Musi być to kolor biały i przejrzysty, ponieważ raz użyliśmy siatki zielonej i to powoduje bardzo duży chaos, ponieważ dla motyli jest po prostu za ciemno i one szukają, przemieszczają się w ciągu dnia za słońcem, tworząc taką wielką kulę. Natomiast przy białej tkaninie rozmieszczają się równomiernie. No i co ważne, w tym namiocie trzeba mieć odpowiednią ilość roślin nektarodajnych, tak żeby motyle miały z czego pobierać nektar. Natomiast po parowaniu samice przekładamy do takich specjalnych woreczków. One dostają tam kwiaty, na które podawany jest syrop robiony z cukru waniliowego i samice składają jajeczka, przyklejając na tkaninę tych woreczków. No i później pod koniec sezonu wszystkie jajeczka są liczone i przykładane do takich szklanych pojemników i całą zimę do następnego roku przetrzymujemy je na zewnątrz w temperaturach no, takie jakie są, tak więc nie trzymamy je w jakichś kontrolowanych warunkach. Udało nam się praktycznie w 3. roku hodowli osiągnąć takie ilości osobników, że mogliśmy zacząć je wypuszczać i prowadzić reintrodukcję. Na początku były to 2 historyczne miejsca występowania, u nas w parku Góra Chojnik oraz rezerwat Kruczy Kamień, jest to Nadleśnictwo Kamienna Góra. W 2019 roku wypuściliśmy w rezerwacie Kruczy Kamień 150 par motyli, natomiast na Chojniku wypuszczaliśmy 300 gąsienic. Monitoring, który prowadzimy co roku, wykazał, że w każdym roku zachodzi, te pokolenie potrafi przeżyć bez naszej pomocy, potrafi cały rozwój przejść w warunkach naturalnych, natomiast nie są to jeszcze takie ilości, które by gwarantowały, żeby ta populacja była stabilna, tak więc co roku staraliśmy się zasilać nowymi osobnikami i tak w roku 2020 wypuszczaliśmy 490 motyli, natomiast w 2021 roku wypuściliśmy 1916 gąsienic i 2529 sztuk motyli. I rok 2022 był dosyć taki dla nas trudny pod względem hodowli, bo u nas to najlepiej się udaje przy ilości około 10 000 gąsienic hodowla, natomiast w tym roku mieliśmy ich 30 000. No i tutaj dzięki porozumieniom z wieloma partnerami udało się wytypować 12 lokalizacji, na których sumarycznie wypuściliśmy ponad 19 000 gąsienic. Natomiast problemem dla nas jest na tym etapie monitoring wsiedlania, ponieważ jest to dużo lokalizacji, tam się trzeba pojawiać dosyć często i myślę, że w przyszłym roku będziemy to rozwijać, bo mamy na to możliwości w projekcie i też bardzo myślę ważnym narzędziem będzie citizen science i tutaj pomoc osób po prostu, które się interesują tym gatunkiem. No i na koniec też chciałem zasygnalizować taką sytuację, czemu w ogóle się zajmujemy tym gatunkiem, po co jest ta ochrona przyrody. Jak to już wcześniej mówiono, niepylak jest gatunkiem parasolowatym, czyli chroni zarówno siedliska, jak i również inne gatunki. I przez to, że jest taki piękny i też on wsiąkł w historię tutaj regionu, ponieważ pojawia się w wielu miejscach, między innymi tam, gdzie prowadzimy hodowlę w Muzeum Gerharda Hauptmana w Jagniątkowie, pojawia się na freskach, tak więc widać to, że był ważny dla tych ludzi, którzy tutaj wcześniej mieszkali. Natomiast wszystkie działania zmierzają do tego, żeby zachować z jednej strony siedliska dla tego gatunku, ale to przekłada się na to, że mamy lepsze środowisko życia dla ludności lokalnej, zachowujemy krajobraz, staramy się rozwijać turystykę, która nie jest inwazyjna dla tego regionu i dajemy pewne możliwości dla społeczności lokalnej rozwoju w oparciu o te aspekty przyrodnicze. A z drugiej strony przez to, że ten motyl jest po prostu taki piękny, to wykupił nam taką ilość czasu antenowego w różnych telewizjach, że gdybyśmy mieli normalnie za to zapłacić, no to by były bardzo duże pieniądze i tutaj te idee ochrony przyrody ten niepylak bardzo fajnie potrafi unieść na swoich skrzydłach. Tak więc dziękuję bardzo.
Thank you, Derek. I think it was very good summing up the whole discussion. Are there any questions to the whole presentation? Yep.
Are you taking questions from the whole morning session? Or okay. A question for Powell about you showed photographs. Sorry, Aidan Whitfield from Butterfly Conservation Europe. You showed photographs of the habitats from 100 years ago when there were very few trees, and now. Is the change passive because of abandonment, or is the change active because of tourism or people planting trees? What has created the difference?
The difference is created by both of factors you have mentioned. The first one was the abandonment of the pasture. When the national park was established, the pasture was taken out. But the second one, important, was afforestation. There was afforestation natural due to ecological succession, but also intentional afforestation, because in several decades ago, there was assumed that natural means forest. So it's the problem.
Okay. Thank you.
In the European Alpine situation, Austria, Bavaria, Alto Adige, it's basically the total abandonment of the alpine meadows. The farmers today, the livestock owners, are hobby owners. They have 10 sheep, 5 sheep, 15 sheep. They're confronted with the return of the predators, which obviously if you have 10 sheep, you cannot do livestock protection. It's impossible. And this coupled also with a dramatic reduction of the animals like goats and sheep. I mean, Austria is missing about 100,000 goats. Sheep do not debush. They don't. They eat grass. Without sheep, we cannot debush. If you cannot debush, you have afforestation. So this loss of habitat will be dramatically increased in the next couple of years, and that coupled with the increase of temperature, which I said earlier, will lead to the forest boundary, Waldgrenze, I think it's forest boundary, of about 200 to 300 meters high up. So either the Apollo learns to climb up the mountain into the rocky faces that are left. But if you walk through those mountains, you see the changes that are just incredible. And there's no way we can get those farmers to bring the sheep and the goats back up there. It's just, it's a hobby business. It's not a commercial business.
Okay, are there any questions?
Hi. So I have a very general question about the reintroductions, about the genetic of reintroductions, because usually we are trying to use genetic data and climatic data to guide the conservation, finding similar populations, genetically similar populations to the extinct ones. But those populations got extinct in the 19th century. So what should we be aiming for? Should we maybe focus on kind of anticipating the changes in climate and future changes in climate and already finding populations that are climatically similar to the conditions that we are expecting on the site? What do you think? Would that be a good approach?
Totally, it's true. We are when we model climatic similarity, we are doing it usually with current conditions, but we should try to model past conditions. I think this is a practice that it's happening already. People is aware of. it. I don't know what's happening. I lost it.
Hello?
Yeah. And another thing that is interesting, we also used to, we need to think about introducing reintroductions in terms of connectivity in the future, because we might be maybe reintroducing a population because it's much more similar to a population that it's much closer, but maybe the population that is much closer but it's genetically more different is going to be in that side in 100 years. So in that sense, if we reintroduce another set of haplotypes, but the other population is naturally going to reach that place, maybe we are more interested to anticipate that and introduce the one that will be climatically similar in the future, even if it's genetically not. So aspects, I don't know, just some thoughts about it. I don't think that's crazy to think.
I tell from the practical point on view. In the practice, there are two ways. The first one, people are insist to prevent the as similar genetic material as it was before. But there is a problem because sometimes the data are unavailable because the material in collection is too old to analyze them. The other approach is just to conserve as well adapted to local as well adapted to the local habitats. And sometimes people do not need genetics for it because they say, okay, the host plant is like that, so the similar subspecies which use the same host plant is here. In fact, the problem is not where the population is extinct very many years ago, because any introduction is in fact safe. The problem is when we try to introduce in the new population close to the old population, because in this case there is the risk of outbreed depression, because we may infect the neighborhood population, but the material introduced. If the species extinct several years ago, we may introduce, in fact, anything.
Okay.
Thank you.
There are two problems going together. One is the genetic structure, and one is the climate change, and that's a question. Unfortunately, Claudio did not print his poster, but we did the climatic modeling for Apollo and shifting this into the past and into the future. And we are not skeptic about the future of Apollo. The problem is because there will be still a huge amount of suitable or climatically suitable areas. The problem is that the areas currently are covered by dense forests, which are not good for that species. But we are happy that we have such events like the forest fires, for instance, in this České Švýcarsko National Park, which will really make a very interesting landscape, maybe even possible for the species. So I think that really here the conservation needs to go directly with all that modeling and just to see, not to be unhappy because of the ongoing climate change, but just to watch for the opportunities.
Okay, any comments on that? Yeah.
Let me quickly reply to this. I think the word we are challenging or have difficulty with in our work is conservation. this work is not about conservation. It is enabling these species to adapt to the rapidly changing environment. If we just conserve, it's what you said, we will go back in history, take that one species which didn't survive in this one area because of everything that happened, and that's not going to be successful. So what we need to do is distance ourselves a little bit maybe from the wording conservation into enabling or supporting these species to adapt to this changing environment that they are facing. And this can be looking at the genetics, looking at which species may be able to survive in this specific habitat, which may not be genetically identical to the species that was there 20 years ago, because the environment has changed. And we are in the middle of this change. So if we want the Parnassus apollo, ... whatever subspecies to survive, we need to distance ourselves a little bit, and we will also be faced with a very tough decision. In most of our work, we like to minimize risk because we are afraid that we upset something. But we are basically sailing in uncharted waters. We do not know what will happen in 5 years, where this thing will go. So there will be some decisions which will be risky, and it will need some people to be willing to take that risk. And we will fail, but we can learn from these failures, and hopefully this species as well, because we should not underestimate a species that, when I looked at your map, traveled from Asia all the way to Europe, that these species can adapt. So I think we are sometimes the problem in how to move forward and should take that little bit of risk.
Yeah, indeed. There's one question.
I have a question about reintroduction places. Do you reintroduce butterflies into private or non-private, some protected places? Because we study other umbrella and flagship species. It's Maculina butterfly, and we quantified some treats and urban pressure in Kraków and outskirts, and we revealed that 2/3 from initial 70 sites was abandoned, and the rest of them was built up. However, built up places are the worst reason of population decline. And we revealed also that protection per se, butterfly protection, species protection per se doesn't work. The only protection that works is protection of sites like Natura 2000 or something like that. The owners and authorities are not aware of they don't want to protect butterflies. That's the question. What do you protect? Butterflies per se, or protected sites with butterflies inside?
W naszym przypadku my skupiamy się w pierwszym kroku na podjęciu współpracy z jednostkami, nazwijmy to rządowymi. Tutaj głównie się opieraliśmy o jednostki Lasów Państwowych oraz o Wody Polskie, ponieważ łatwiej nam zorganizować tą współpracę, kiedy partner jest przewidywalny. Możemy tutaj podpisać odpowiednie porozumienia i zaplanować w dłuższej perspektywie te działania. I staramy się jeszcze tak organizować tą współpracę, żeby właściciele czy zarządcy danych obszarów nie ponosili z tego powodu jakichś kosztów. Czasami te ustalenia są bardzo proste, bo na przykład polegają na zmianie terminu koszenia wałów przeciwpowodziowych. na przykład na koniec sierpnia i wtedy my wprowadzamy wcześniej gąsienice, które korzystają z roślin, które rosną na wałach, przechodzą pełen cykl, samice później składają jaja, no i w tym momencie wchodzi zabieg, który no zawsze będzie się na takich obszarach pojawiał, czyli koszenie. Tak więc oczywiście to może powodować trochę zmniejszenie na tych konkretnych punktach liczebności, ale my też musimy takie rozwiązania testować. I w przypadku akurat też Pylaka Apollo próbujemy wchodzić blisko ludzi, bo te niektóre powierzchnie są wprost jak gdyby obok miasta, tak więc te spoty staram się zakładać, żeby one z jednej strony były w miarę blisko siebie, żeby te motyle mogły migrować, ale też żeby miały jak gdyby kontakt z człowiekiem w tym sensie, żeby pokazać, że ta dzika przyroda jest wokół nas i mamy wiele też takich później, bo robimy to celowo w drugą stronę sytuacji, że się pojawiają podmioty prywatne, właściciele jakichś gruntów, którzy chcieliby wtedy przyjąć jak gdyby te nasze motyle czy gąsienice na swój teren i się dzwonią, pytają nas, czy tu są dobre warunki, żeby na przykład tego motyla wypuścić, ewentualnie co powinienem zmienić w swoim gospodarowaniu na tym obszarze, żeby ten motyl mógł się tutaj pojawić. Tak więc to było przemyślane, żeby zaczepić się najpierw z pewnymi partnerami, a później promować to wśród prywatnych, bo bardzo duża ilość tych lokalizacji jest na gruntach prywatnych.
So just the last comment maybe, because we will use the time for the coffee break.
You know, it's obvious that butterfly may be protected only together with the habitat, because protecting without habitat, it's for sure it failed. However, you know, the Kraków and Makulina is toughest example as possible, because this Makulina lives in extremely attractive for urbanization area. With the Apollo butterfly, we have no such problem, because in Pieniny, this habitat suitable for Apollo butterfly is also unsuitable for the economic activity, and sometimes there are private localities like Długa Grapa is private, as well as Zamczysko. Yes, so some of localities are private, but there is no conflict of interest, so it's easiest than in the Krakow.
I would like to come back to the genetic discussion. When you release most similar individuals to the extinct population, it means that you release individuals which are best adapted to the local conditions. But conditions change, so maybe it's a better strategy to release a population with a high genetic variability and a high ability to adapt to changing situations, especially when you deal with insects with a high number of offsprings, a short generation cycle. If you deal with vertebrates, it's much different. So maybe it's better for a reintroduction to build up a very population with a high genetic variability, that the population can adapt by itself to changing conditions.
There is two problems with this idea. The first one is very specific. The changes of habitat for Apollo butterfly are in the many cases they are rapid and human driven. When we have the forest in the place where the pasture, Apollo butterfly didn't survive in this area. So in Pieniny, the restoration of Apollo has the two main activities: introduction and habitat restoration. Moreover, if we would like to have this umbrella effect of butterfly, when we introduce the subspecies of population which is not adapted to this particular habitat condition in this place, the umbrella effect may fail because another species will not be protected. And moreover, there is all the time risk of this outbred depression. So as the Gerard said, each case should be individually analyzed.
Yes, I agree. I just would like to point out that when we talk about the most genetically similar, we are usually still blind. I mean, because we don't really know. We would need very deep genomic screens or scans to really know what is the most genetically similar. It might look genetically similar for the markers we are investigating, which are usually mitochondrial. Then if we go to genomics more deeply, maybe we would find some other patterns. So whatever we do, we're a little bit blind, and I just want to stress on having the opportunity to really screen better the genomes before deciding what is genetically similar or not. It would be very, very much desired for introductions. Yeah.
Okay. Thank you very much. I would like to invite you all to the coffee break. There will be also still some space and time for discussion later on, so keep your questions in mind, and we will also ask you to ask them later. And the next plenary panel, it's like 11:45, so almost half an hour break.